How are you navigating the challenge, or what advice do you have for our audience members who may be navigating the challenge of pre-existing immunity to adenoviral serotypes, particularly considering prior exposure, widespread vaccine deployment? What do you think?
So I'm going to be very boring. This is the intratumoral delivery for us has been the solution. So we have a large body of evidence generated both in preclinical animal models as well as in the clinic, whereby we see that if you deliver systemically the adenovirus, there is a very fast development of antibodies, not just for patients that had pre-existed immunity to adenovirus. But the adenovirus is extremely good at inducing the immune response. That's what we leverage actually as a part of the mechanism of action. But if you deliver it intratumorally, this is not the case. And in animal models was extremely clear, even after repeated injection, you don't induce such a strong immune response against the adenovirus. In fact, we've been able to do it up to 3 injections. We've been able to deliver this vector to the pleura, where there is a massive amount of blood flow and there is a lot of accessibility for potentially neutralizing antibodies. And we've seen that there is no correlation between pre-existing neutralizing antibody to AdenoV5 and biological measures of the outcome of the delivery of the virus or clinical outcome. So it's extremely clear that when you deliver locally, you have this activity, because the activity is mainly linked to this oncolytic capability, the capability of a transiently derived transgene, the TK transgene, as Karen mentioned.
Yeah, and if I can add to that too, in addition to that, if it is something you want to use a little more broadly, instead of very locally where it may not make a difference, there are other serotypes available of adenoviruses that people have worked with. And so that's one way to potentially reduce the pre-existing immune responses.
Great. Okay. And Karen, while I have you, so what technological or design advances are helping overcome immunogenicity and toxicity hurdles associated with adenoviral platforms?
Yeah, so the main things have really been to remove more and more of the viral genes. So the original vectors only had a few small deletions, and you can remove more and more until you get to what are called gutless vectors. And so that does seem to be helpful in reducing immune responses and toxicity. So I'm not talking, when I say immune responses, I'm not talking about neutralizing antibodies. I am talking about cell-based immune responses to its being introduced there. So that does help. It does make it a little bit harder to manufacture, but it is, I think it is doable. And certainly, you know, if you add to that potential modifications to the capsid proteins, you can improve, capsid proteins, you can improve tropism. So, so it does help to remove more and more of the adenovirus genes so that they're not expressed even at small amounts when you think you've inactivated it.
Sure, sure. Francesca, I'd like to get your perspective on this as well. You know, what technological or design advances are helping overcome immunogenicity and toxicity hurdles?
Yeah,
with
I think
regard
that
to
this is, I think that in the adenoviral space, Karen is right. I mean, you can strip the virus as much as possible, but with the adenovirus, it's such an immunogenic vector that you ended up losing some key properties when you do that, and then it becomes just a tool for delivery. And so I think that it depends very much on what you want to achieve. If you want to achieve a systemic immune response or a local immune response against the tumor, I think you want to retain that and maybe work on the delivery methods. There has been some ways, for example, CG Oncology, when they deliver their adenovirus, that's a replication-competent vector, to the bladder, they sort of pre-treat the bladder to favor more integration. They remove some of the mucosal barriers, and so they limit the biodistribution. They favor the biodistribution in a short period of time that limits then the exposure to external part. I think that in general, also the replication competent versus defective. This is a modification that is often made, and it changes completely the biodistribution, changes completely the safety profile. In general, we like to leverage the immunogenic properties of the vector, and so we don't want to, we try not to do that. We try not to make these modifications.
Sure. As noted in our second poll question, and as the results came up, it sounds 37.5% of our audience are quite concerned about manufacturing scalability and cost as a barrier to advancing their preferred delivery platform. And so how would you say adenoviral vectors compare to lentivirus, LNP, what have you, in terms of scalability in manufacturing, especially for large scale or commercial production? Francesca, perhaps we could start with you.
I've got not much experience with lentiviruses. I can tell you that the adenovirus is a fairly easy vector to make. I mean, there is a lot of knowledge around that. The cost of goods actually are very limited. We've seen how it was able, how we were able as a, in England, the first adenovirus with the first vaccine for COVID was actually adenoviral based, and it was able to produce it with a very low cost in a very short period of time. We deliver, 10 to the 11th viral particles per 5 times 10 to the 11th in each injection. So it's a fairly high titer, and we obtain this high titer fairly easily with scalability. That is different for other vectors. HSV has got a completely different manufacturing process. For the lentiviruses, from colleagues that work in the space, is a little bit more complex, but I leave Karen, this is more of expertise to answer this question.
I was going to say to Karen, please, share your thoughts.
Sure. So I agree with Francesca that adenovirus is relatively easy to make, to produce in large quantities. I think the one thing which, when I'm finished with this part, I'd like to hear your comments on are also whether, as you scale up, whether replication-competent adenovirus becomes more of an issue, or whether it's, how straightforward it might be to prevent that from coming through, because there are levels at which you want to keep it below certain levels. And so you do have to test for that. With these other processes, they tend to have additional steps involved in them, right? So lentivirus, again, you produce, each time you produce it new with a transfection system. With LNPs, also, they're produced each time new, but the difference there is that you're not depending on mammalian cells to produce your virus for you. So it is more of a chemical reaction. I think of that naively as somebody who deals with biologics all the time. But then once you form your lipid nanoparticles, you have all the components of it, you then need to encapsulate your payload. So whether it's RNA or something else. And so there, because the technology is a bit newer, I think there's still some advances in understanding with LNPs there, how to standardize it, and purity and reproducibility. So that's kind of where they stand. The differences are. Francesca, I don't know if you want to comment on the replication competent.
Yeah, you raise a big problem in the space of adenoviruses. So the replication competent adenovirus is almost innate to the production, is an innate problem to the production of adenovirus. So basically what happens is that as you produce your replication defective virus using modified cell line, you ended up producing some replication competent vector that classically is almost the native form of the vector, and it doesn't have anything, it doesn't carry your transgene of interest and so on. There is a lot of literature around that, how to modify. We've been working on it, and we have a great CMC team that has really worked hard on a couple of strategies. One is using a cell line as a master cell bank and master viral bank that is quite early in the development, and so has very little RCA, this replication competent vector, to start with, and then modification that you make to the methods. It's very classical, the formation of RCA, especially with old type of cell banks that they've been using in the past. but new methods of purification, new methods of expansion can contain these numbers. So we've actually been extremely successful in maintaining this number of replication-competent adenoviruses low. And so, and that's great because it's one of the requirements that the FDA has.